This is the Chapter 12, Nutrition and Energy commentary. Continued commentary. A few more energy terms you need to be familiar with. Metabolism. Metabolism is the sum biochemical reactions occurring within an organism. So the sum energy processes within an organism. We break these up into two parts, catabolism and anabolism. Let's start off with anabolism. One little mnemonic trick we know is whenever you think of steroids and bodybuilders, the first thing you should come to mind is anabolic steroids. So anabolic means to build up. The fancy word for build up from smaller particles is synthesis. In layman's terms, it means to build up. So anabolic is, or anabolism is the sum energy reactions to build up biomolecules. So when we're building up new, larger molecules from smaller ones, such as creating the insides of a brand new cell. Catabolism is the opposite. That's when we're breaking down. So I always think catabolic, cannibal, we're breaking down biomolecules in order to release energy. You're responsible for those two terms. If that helps you out, great. If it doesn't, then, well, make sure you know. An array of chemical reactions that are organized into an orderly, well-regulated sequence are called metabolic pathways. So these are established back-to-back chemical reactions that occur over and over and over. These back-to-back chemical reactions often have enzymes to help speed them up. So again, a series of steps that convert some reactions to a product over and over and over. So for example, we have the citric acid cycle and the electron transport cycle. I'm going to go ahead and go to Chapter 13 really quick just to give you a point of reference. So here's an example of the Chapter 13 handout. This right here is a metabolic pathway because it's the same reaction steps over and over that always occur. And we often have enzymes at key locations to help speed up this overall series of reactions. All right, let's go back to Chapter 12. We're going to first focus on catabolism. That's when an organism cannibalizes old biomolecules to release energy. So the first stage of catabolism is digestion. We break down the larger biomolecules, fats, carbohydrates, and proteins into their smaller building block units, such as fatty acids, glucose, and amino acids. So this is what occurs within your stomach when you first eat that piece of chicken, for example. In your stomach, acids and enzymes help break it down into amino acids. Stage 2 is the production of acetyl-CoA. So regardless of whether we have a triglyceride from fats or a simple sugar from carbohydrates or an amino acid, all three of these can be broken down into a very important energy molecule. This energy molecule looks something like this. This has two carbons, and on this carbon right here, there is a carbonyl group. We often refer to this particular structure as a acetyl group. Now, there's an extra bond on this carbon right here, and depending on what it's attached to, that's where you might have seen this particular name before. So, for example, in the organic section, we talked about acetic acid. the common name acetic acid. And that refers to the fact that there's an OH here, so this is an acid, so acetic acid. Or if I were to sit there and change it to a sodium, then you might have seen a sodium acetate. My point in this is that whenever we have this particular structure right here, that's the acetyl group. In biochemistry, often what happens is we'll attach it to a sulfur and a really, really big enzyme, coenzyme A, which we summarize as CoA. So oftentimes we'll write it like this, acetyl-CoA. So it refers to the acetyl group and then CoA. It turns out that all three of the three classes of biomolecules can be broken down into the acetyl group. The acetyl group is then pushed into the citric acid cycle. That's the metabolic pathway, and it is the steps are cyclical in nature because a reactant forms a product and it goes all the way around, forming a brand new reactant that can continue on. So all three of these are broken down into the acetyl group. The sulfur and CoA is actually you can think of it as a shuttle that transfers around the acetyl groups to the desired location. It's the acetyl group that actually, however, goes in to the citric acid cycle. And in the citric acid cycle, various chemical reactions occur, and the purpose of these reactions is to release energy to reduce NAD and FAD. Now, most people think that the citric acid cycle powers the formation of ATP, and they're half right. The steps are actually more complicated. It actually releases the energy, and it releases hydrogens in order to reduce NAD and FAD. So what it does is it basically donates hydrogens, which is why we get the term reduce. Remember, oxidizes to add oxygen, reduces to add hydrogens. In this case, we're going to go ahead and add hydrogens and reduce it. So it forms a NADH. And I really should make this an A, by the way. Okay. And this other one, FAD, it forms a FADH2. So, again, it releases energy to reduce these special biomolecules NAD and FAD so that they form NADH and FADH. It's these intermediate molecules which power the formation of ATP, and that's in the electron transport and oxidative phosphorylation steps. So, what we're now going to do is we're going to focus on what the individual biomolecules look like. I'm going to ask you to recognize these biomolecules, although I'm not going to ask you to sketch them. So, let's go ahead and start going down the line. In the first stage, let's go back a bit, I guess. Stage 1, again, we're digesting the large classes of biomolecules, proteins, polysaccharides, fats. We're digesting them using enzyme-induced hydrolysis to break them down into their polymer building blocks. Stage 2, the building blocks are broken down even further to form the acetyl group, which is this right here. Keep in mind this S-CoA is nothing more than a shuttle which temporarily bonds to the acyl group to shuttle it to the location it's supposed to go to. So this is the acetyl-CoA. Don't worry, we'll show you what this looks like in a second. Now, in stage 3, the common catabolic pathway extracts energy to produce ATP. So, all this right here, going back to slide 13, is part of stage 3. The first part is the citric acid cycle. That's the metabolic pathway. Again, it's a series of repeated chemical reactions. Here is the other part of the stage 3, and this is the electron transport and oxidative phosphorylation. And the end result is to recharge the ADP to ATP. So, let's go ahead and move on. Here is a picture of ATP. So, here we have adenosine triphosphate. I do want to point out the word adenosine comes from the fact that we've stuck together a adenine base and a ribose sugar. And it formed what the new biochemists common name call it adenosine. So adenosine. So think of this as a special word that is plural, which represents adenine and ribose together. A plural noun, if you will. And then triphosphate, this is the triphosphate, which we've studied before. Now, when ATP is using up its energy, basically it's hydrolyzed at this point and it loses its ester phosphate in a hydrolysis reaction. So here is the slide again. This is actually the same slide that you've seen before, so this should be nothing new. Here's ATP. Water comes along at the right time to break the bond. Let me change to a blue pen. To break the bond, and it goes from ATP to ADP because there's only 2 phosphates. And then this is floating around and energy is released at the same time. Oftentimes we write phosphate, instead of writing out its whole structure, we'll either say PO43-. Biochemists get even lazier and sometimes they'll just write P with a lowercase I. And that's a phosphate. The 3 stages of catabolism are used to provide the energy to go the opposite way. So that way, if you have an old used ADP, then the 3 stages were used to release energy, so that way we could put the phosphate back on. So in that case, we're going to be going the opposite way, ADP to ATP. Here's more math. In this case, I've circled the key reaction here. In this case, ATP plus water forms ADP. And here, physical chemists have actually identified how much energy is released. So basically, 7.3 kcal of energy is released per mole of ATP. It turns out that sometimes in nature will release more than 1 phosphate, in which case more energy is released. So in this case, if I start off releasing 2 phosphates at once, then even more energy is released. So if you notice, when 2 phosphates are released, it creates another type called AMP, which is adenosine monophosphate. Adenosine monophosphate is also used as a activator in a metabolic pathway. So it can be used as a modulator to regulate a pathway. So know that, know this, and let's move on. Oh, here's a nice summary of ATP to ADP used for biosynthesis. That's what we're going to focus on most at this point. And then the 3 stages of catabolism are used to recharge the ADP to ATP. So know that. Know what a hydrolysis reaction. It's a reaction that requires water to break or cleave the bond. So in this case, hydrolysis reaction. And then know the general reaction I have here, and also know that AMP can also be used as a modulator. All right, now when we're actually forming ATP, it actually occurs in the powerhouse portion of the cell called the mitochondria. The mitochondria actually has two layers, and it has a inner membrane layer and a outer membrane layer. Now, if you notice in this picture right here, you have a outer membrane and then you have a inner membrane. Notice in between you have a inner membrane space, and then if we're looking inside the inner membrane, you have what's called the matrix. So memorize all of these different names. It turns out you have multiple folds on the inside. So at this point, we're just going to call them the crista. It turns out that the more surface area you have, then the more ATP formation you can have, because there's a lot of important embedded enzymes into the fold. So oftentimes it looks very folded, like it is right here. So know the general parts of it, because we're going to get into the citric acid cycle, and we'll get into the electron transport chain. And the electron transport chain is really important if you understand where you have all the location and terminology of the parts of the mitochondria. Let's move on really quick. This is what a coenzyme A looks like. So in stage 2, they all broken down into acetyl groups, and they're shuttled around with coenzymes. This right here is the acetyl group. And then the rest of this giant monster, that is the coenzyme A. Think of this right here as a shuttle, which transfers around that tiny little acetyl group. And then basically it drops off the acetyl group to the desired location, and it picks up a new one. It forms a temporary sulfur bond to the carbon, which we call a thioester bond. So let's go ahead and move on. And these coenzymes, we often get these from our diets in the form of vitamins. In this case, coenzyme A is actually rebuilt from another type of vitamin, pantothenic acid. So let's go ahead and move on here. This is what NAD looks like. NAD and FAD, these are the intermediate energy molecules which are reduced. They pick up hydrogens, and they then go and help power up the electron transport chain, which is used directly to make ATP in the phosphorylation step. So in this case, it's nicotinamide adenine dinucleotide. We get the term dinucleotide because we think of this as two nucleotides which are combined at a phosphate inorganic ester linkage right here. So in general, you can think of a nucleotide as having a amine or amine-related amine derivative. You can think of it also as having a ribose sugar. The sugar can either be cyclic or it can be open chain, as long as there's 5 carbons and it's a ribose. And then of course we have our phosphates. Because there's 2 of these linked together, this is where we get the term dinucleotide. And in the front, the way they've designed it, on one nucleotide, we have the name of one of the amines. On the other nucleotide, we have the name of the other amine. So the top one is nicotinamide, the bottom one is adenine. I do want to point out on these that if you see right here, this is the only place where hydrogens are going to be bonded to. So in this form, it's just NAD. However, this pi bond, when exposed to a hydrogen that's going to bond to it, it'll bond right here. It'll cause a set of chain reactions where when this bonds to it, this pi bond will come down here, and this pi bond will come up and form a brand new lone pair. But in order for this to occur, you need a hydrogen donated and you need electrons. I focused on the hydrogens in this class, but yes, you also need a hydrogen and you need 2 electrons in this case. So that way, the hydrogen and the electron pair can form a nice covalent bond, as you see here. Please recognize this molecule. Now, when we get into the later part of the course, we'll talk about how energy is released for ATP synthesis when oxygen is reduced by accepting electrons and hydrogen ions to make water. At this point, you don't have to know it for the upcoming quiz or exam 4, but I do want to point out that we're going to talk about this in chapter 13, part 2, I believe, as well as chapter 13, part 1. So what you need to know is NAD is converted to NADH, and hydrogen and 2 electrons, these are given to it in the citric acid cycle. So the whole purpose of the citric acid cycle, in a nutshell, is to go ahead and protonate the NAD so that it forms NADH. To give it hydrogens and electrons to form NADH. And this is a molecular middleman which will eventually help power up the other process to give it to regenerate the ATP to ADP. So understand the general idea here, but I'm not going to test you on this portion for the upcoming quiz or exam, only on the final. And you can go through this, but again, I won't test you on this until we actually get to the quiz and final. Here is, so if we just focus on this part right here, on slide 22, we see the before and after. So here's the before, where it's just NAD+. Notice the plus comes from the fact that there is a ammonium present that in this heterocyclic amine. So, and it has a plus charge. You then have 2 electrons and a hydrogen donated from the citric acid cycle, and it forms NADH. Why? Because there's a brand new hydrogen and 2 electrons in the form of a covalent bond that are now attached. See how it manipulated the pi bonds? One pi bond came down here, and the other one came right up and formed a brand new lone pair. So it acts as an electron acceptor, aka it is reduced. We never talked about electron acceptor. We always talked about oxidation and reduction as relating to oxygens and hydrogens. So if you want, you can scratch that out and say it gained hydrogens. Here is flavin adenine dinucleotide. This is also a dinucleotide because here is one nucleotide. Here is another. Yes, this is a ribose. Notice it's a open chain ribose, but technically it's still a ribose. So it still fits into our definition of nucleotide. So the first part we have a flavin. This is the flavin. And it's derived from the ADP and the vitamin riboflavin. Over here we have adenine. That's where we get the name. So I'll give you some insight into this. Recognize. Notice in blue, FAD is going to be reduced and pick up 2 hydrogens. It's also going to pick up 4 electrons in order to form covalent bonds to those hydrogens. It then forms a FADH2. So in this case, here's before. Here is after. Now, you can either give me the full name, you can give me the acronym, and both would be acceptable on a future quiz or exam. It's often involved in the oxidation of alkanes to alkenes. We often see this in when we start getting into fatty acid synthesis. That's the very last chapter, chapter 14. And then this also can play a part in the electron transport chain and the ultimately the phosphorylation to produce ATP from ADP. So bottom line is, again, this is a molecular middleman. The citric acid cycle is the one that is responsible for donating the 4 electrons and 2 hydrogens to form FADH2. And this will power up indirectly in order to form ATP. So you have 2 uses for these. Let's go ahead and I'm going to go ahead and end on this slide right here, just so it gives you a point of reference. This is the citric acid cycle. So here is the acetyl group right here, transported by the shuttle CoA. And what it's going to do is it's going to donate that acetyl group into a larger biomolecule and add it together. At which point it starts breaking down the biomolecule. As it does this, you'll notice there's NADA. There is NAD, and guess what happens? It gets to pick up a hydrogen and is reduced. Notice down here, here's another example. Another free NADA gets to pick up a hydrogen, is reduced. It also picks up the electrons, don't forget that. There are other weird biomolecules we haven't talked about, so we may address them at the very end of class, but I'm not going to test you on them. So GDP to GTP, that's something different. Here's the FAD to FADH2 that I talked about. Notice this one picks up 2 hydrogens instead of 1, along with 4 electrons. So as you can see, the main purpose of the citric acid cycle, it doesn't produce ATP directly, rather it generates NADH and FADH2. And these will help facilitate the process of producing ATP, and we'll get to those steps at a later time. So going back here, you're responsible for the general function, and you're responsible for recognizing the structures. And this concludes this set of slides.